Have you heard what's happening in Calgary, Canada? Home to some of the world's best researchers and innovators in life sciences, Calgary is advancing healthcare solutions to solve global challenges. Calgary's dedication to the life sciences sector is evident in its labs, hospitals, schools, and the minds of its people, with its top institutions producing internationally recognized research and more than 110 life science companies backed by a highly skilled pool of talent. The life sciences sector is accelerating innovation in Calgary. If you're a bright mind or a bright company, Calgary is just the place for you. Take a closer look at calgarylifesciences.com. I'm Daniel Levine, and this is the Bio Report. The body has a natural cellular recycling machinery known as the ubiquitin proteasome system that breaks down unwanted proteins. Kymera Therapeutics has developed a drug discovery platform that exploits this natural biologic process to target disease-causing proteins that had been previously considered undruggable using small molecule therapies. We spoke to Nello Mainolfi, co-founder, president, and CEO of Kymera, about the company's discovery platform, how it exploits a natural house-cleaning mechanism within the body, and why this approach could enable the targeting of proteins that previously had been considered beyond the reach of small molecule therapies. Nello, thanks for joining us.
Danny, thanks for the invite. Great to be here.
We're going to talk about Kymera, its ability to degrade proteins in a targeted way, and the implications for being able to do this. Perhaps we can start with the ubiquitin proteasome system. This is a natural housekeeping process within the body. What's its purpose and how does it work?
So great question. First of all, actually, the discovery of how the ubiquitin proteasome system works was the subject of a Nobel Prize, I believe, in 2004. So it's a science that has had a tremendous impact on how we understand cell machineries and disease and health and homeostasis. So the way that the system works is by recognizing proteins that are either misfolded or accumulated or they are kind of in the wrong concentration. And the ubiquitin proteasome system uses this series of protein cascades that start with E3 ligases that form complexes with protein of interest and eventually leads them through to proteasomal degradation.
Well,
go ahead.
You mentioned the E3 ligases. These are enzymes. What exactly do they do, and how do they work?
So E3 ligases are, I guess, a class of proteins that, again, are part of the ubiquitin proteasome system. There is actually about 600 E3 ligases that have been characterized. And their role is to recognize these proteins that we're talking about, usually through a protein-protein interaction. So they recognize that protein, they bind to it, and then through this spatial proximity arrangement, they bring the rest of the UPS system to this protein that is misregulated, and then that leads to that protein degradation.
So how are you able to exploit these with your platform, and what does that accomplish?
Yeah, so the beauty of targeted protein degradation, which is the technology that Kymera uses to generate what we believe will be transformative medicines, is so the beauty is the simplicity of the concept. So we just described, hopefully somewhat clearly, what the ubiquitin protein system does, which again is recognizing protein and degrading them. So what targeted protein degradation does is introducing small molecules. We call them degraders. We call them heterobifunctional degraders. And the small molecules' role is simply to generate an interaction between the ubiquitin proteasome system via binding to an E3 ligase, and at the same time binding to a protein that we believe is disease causing. And so this interaction that is facilitated by our heterobifunctional degrader leads the ubiquitin proteasome system to then recognize this protein as one that needs to be destroyed. And so then the cell machinery does the job. We just bring the disease-causing protein to the proteasome. That's really what we do with the technology.
So typically when a protein that's involved in a disease state is identified, a drug company would try to develop a drug that might inhibit that protein by binding to it. I take it one of the problems is that there are many serious medical conditions that are considered undruggable because there's no way to get a small molecule to bind to them. Is that correct?
Yeah, that's a great point. Yes. I think maybe if I take it one step back, we have our proteome, which is obviously encoded by our genome, is made of 20,000 proteins. And I think what is fair to say that only about 15 to 20% of this proteome has been drugged. And one of the reasons is because we really didn't have the tool to drug these proteins. And so one way that we believe is going to revolutionize how we think about drug development is using targeted protein degradation. And the point that you were making is that with these molecules, we actually don't have to inhibit the function of the protein. We just need to bind to it. So naturally, the opportunity of addressing a novel class of disease-causing proteins is enabled by the fact that we are not constricted by this inhibitory mode, and we can just go after any protein that is in the cell to which we can bind.
So just to finish the thought, how does this get you to treat conditions that might not have been treatable through conventional small molecule drugs?
Yeah, I would say not only conventional small molecule, but I would say any other modality. So the reason why this is now accessible is because, again, as I said, there are many validated disease-causing proteins, pathways, and targets for which we know either human genetics or we have preclinical strong pharmacology or we have had some other pathway clinical proof of concept, but against which we don't have an opportunity to develop a therapy. And, one example could be that it's a protein that a small molecule cannot inhibit. But just to give you a sense of how broad the solution can be, let's say it's also a protein that is in the cell, so an antibody therapy cannot get to. It's also a protein that is involved in a disease that is systemic. So obviously, for example, oligo-based therapeutics like RNA or CRISPR gene therapy maybe cannot address because they cannot be delivered systemically, but only in particular tissues. And so the solution is not just for small molecule, but the solution is for actually any protein that any other technology is not able to tackle. And then again, just to clarify, any protein that is in the cell that a small molecule can bind to but doesn't have to inhibit can be targeted with this technology.
So walk me through how your platform works. How you've identified a protein that you want to target. How do you get from there to a drug candidate?
Yeah, so it's so once we get into the details, it's obviously much more nuanced than the simple concept of binding protein and binding E3 and degrading protein. So the first thing that we do once we've identified a protein that causes disease. And one example could be our IRAK4 program. We know that IRAK4 is responsible for signaling inflammatory signals that generate pro-inflammatory cytokines and chemokines. So we know that in many patients, the upregulation of this pathway and of this target causes broad inflammation, whether it's rheumatoid arthritis, whether it's skin inflammation, whether it's GI inflammation. So once, let's say, we identify a particular protein that we believe is a node that creates pathology, and we decide that we want to remove it, then what we do is ask the question, is there a small molecule that has already been identified that is binding to this protein, or do we need to find the small molecule that binds to this protein? So that's one question. Then the subsequent or parallel question is, what is the right E3 ligase that we need to deploy to degrade this particular protein? And the question arises because we need to find an E3 ligase that is expressed in tissues, in cells, in organs where this particular protein is responsible for disease. And so we need to, and not all E3 ligases are expressed equally in all tissues and organs. So first, do we have a small molecule to the target? Then do we have the right E3 ligase? Then the third question is, do we have a ligand to that particular E3 ligase? Meaning, do we have a small molecule that binds to that E3 ligase? Once we answered all those questions and we have the right tools, then we build this heterobifunctional molecule. We basically link the small molecule that binds to the E3 with the small molecule that binds to the target, and we make these two small molecules into one, as we call it, heterobifunctional degrader molecule. And then through, obviously different and several iterations of optimizations, we get to the drug candidate.
And I understand the name of the company has its roots in kind of describing that. Can you explain that?
Yeah, so that's a great question. So the name Kymera is actually coming from the concept of chimera, which normally is Chimera, which is the mythological double-headed monster in Greek mythology. And so the association with the technology is the fact that these molecules are heterobifunctional, so that in a way they have two heads, just like this mythological figure. The reason why then we ended up changing the CH to KY was, first of all, we wanted to have a less obvious kind of name that would be a bit unique, but also comes from actually the biochemistry of the UPS system, where when we take an E3 ligase that recognizes a protein of interest, there is a transfer of a small protein called ubiquitin, and the small protein is responsible for tagging the protein target for degradation, and this ubiquitin actually attaches onto a small amino acid called lysine. So the code for lysine is actually K. So that's why we decided to change CH to KY. And so the K represents the amino acid that is really the basis of the technology. And then Kymera is, again, going back to the double-headed mythological monster from Greek mythology.
We talked about this in the context of going after previously undruggable targets, but it strikes me that if you can do this with a small molecule, degrade a targeted protein, that it's also a way of developing an oral therapy for conditions that previously can only be treated through things like biologics. Is there a strategy here to develop oral versions or oral formulations for conditions that today are treated by biologics or other costly approaches?
I would say that the strategy is to develop therapies for both diseases and naturally against protein targets that have not been drugged or drugged well before. So I wouldn't say necessarily that it's swapping a biologics for a small molecule, but I would say that it's an opportunity to go beyond the biologics, to go at the core signaling pathway that are responsible for disease. An example, again, going back to IRAK4, we can actually go back to even STAT3, which is the other program that we've publicly disclosed. For IRAK4, lots of therapies go after extracellular cytokines. Again, these are inflammatory molecules through biologics. So you inject an antibody protein to patients that block these cytokines. But in reality, all these cytokines, there are many for which usually biologics are only addressing one of them. All these cytokines signal through this intracellular pathway. So not only with an IRAK4 oral degrader, we're blocking this signaling intracellularly, but we're blocking the signaling of multiple cytokines. So rather than swapping one antibody for one oral small molecule, we're actually going for replacing a series of antibody therapies that cannot be injected in a single human together with one oral small molecule that has the power of multiple antibodies. That's really the opportunity in this case for this program, but also for many other programs that we are working on at Kymera.
Your lead candidate is KT-474. This is in early stage clinical testing. You've had what you've said is a proof of concept. What's known about its safety and efficacy to date?
Yeah, so our IRAK4 degrader program has actually been the first placebo-controlled randomized phase 1 study in healthy volunteer that these molecules across the industry have ever been put into. So Kymera was responsible for, again, the first time this type of study. And the reason why we decided to take this route, because we are a company that believes strongly in understanding the relationship between PK, between PD, which is a downstream, again, it's an impact on protein level, between the protein level that we're degrading and a disease-relevant downstream biomarker. And only if you do a study that is placebo-controlled or randomized, you can actually build the confidence in later development. So what we've learned so far, that we have a molecule, so far we've only disclosed the single ascending dose study. So we've actually shown data after just a single dose. And what's amazing from those data is that we were able to show that with a single dose of our drug, KT-474, we were able to degrade IRAK4 systemically up to 96%. So basically, we had a total degradation of the protein in the body. And that degradation after a single dose was maintained for up to 7 days. So basically telling you that this mechanism is not only very potent, but also very profound, and there is also a time component to it. What we also learned that by degrading this protein, at least 85% or more, obviously we have different doses that give different effect, we were able to show an impact on downstream disease-relevant biomarkers like those cytokines and chemokines that I was telling you before, up to almost complete inhibition on some of those cytokines and chemokines. We've reported 97%. So, and all these profiles, so as I mentioned, almost complete knockdown or degradation that lasted for multiple days, that showed an impact on a wide variety of disease-relevant biomarkers. All of these was really well tolerated by our human volunteers. In fact, there were no serious adverse events that were seen in the course of the study, and we've dosed up to 57 healthy volunteers in this study. So this is also a large number of subjects that were dosed.
I know the company has announced a number of partnerships. Is this an unpartnered an unpartnered asset, or is this part of a collaboration?
Yeah, the great question, Danny. So this KT-474 right now is being developed by Kymera, but we have a co-development partnership with Sanofi. So at the end of phase 1, which again Kymera is still driving, then Sanofi will work with us in phase 2 and phase 3 and commercialization of this drug, and we will be splitting 50-50 the profits of this drug in the US.
Well, you've got a partnership with Sanofi. You also have one with Vertex. I did want you to walk us through both of those. Start with Sanofi. What is the broad parameters of that collaboration?
Yeah, maybe before I do so, given that we went from the real technicality of the science to the programs, maybe just take a minute and share with you what is the long-term vision for this company. So we founded the company in 2016 with the real goal of building a fully integrated global company that will take protein degradation from an early stage that was when we founded the company into a commercial stage engine that would be able to discover, develop, and commercialize degrader drugs. So when you embark on this journey, you obviously have to recognize that there are opportunities where you can try and accelerate that growth through partnerships. So we look at those partnerships opportunity not so much as a financial mean, and in fact, I would say that luckily, at least so far, we've been blessed and lucky to have access to capital in a pretty de-risked manner. But we've looked at these partnerships as a way to accelerate our growth in areas that maybe independently we wouldn't be able to do so quickly. So our partnership with Vertex, for example, that we signed in 2019, gave us an opportunity to move in disease areas that were beyond the ones that we were working on internally. In fact, internally, we're focused on oncology and immunology. With Vertex, with which we have a 6-program partnership, it's all in disease areas that are all outside of oncology and immunology. In fact, there are actually 5 different disease areas. And so what this did for us, again, besides the financial, there was a generous upfront of $70 million. There is $1 billion plus of milestones. But really what that did for us was allow Kymera to be a disease-agnostic protein degradation company really early on in our life, in a way that probably on our own we wouldn't have been able to do. With Sanofi, very different collaboration, but again, fulfilling the same premise, which is with Sanofi, we had a very exciting asset, KT-474, which continues to behave really, really well, as I just mentioned. And because we believe that this drug has the potential to be a best-in-class small molecule oral drug for immunology and inflammation, we realized that in order to compete with large companies that have, drugs in this area, we needed to have a large investment very early in clinical development. And so with Sanofi, we have an opportunity to initiate multiple phase 2 studies and phase 3 studies in a de-risked manner, meaning in parallel or staggered, and invest a substantial amount of money as well as people and operation and commercial infrastructures at a stage where Kymera is still building some of those, and we are in some cases in the early stage of building some of those. So the partnership brought us, again, financial incentives, and I think it was $150 million upfront, and I think we had $2 billion plus of milestones. But so while those are very important and critical, the ability to be competitive and to advance very aggressively in the clinic and to get this drug to as many patients as possible, as soon as possible, was the key strategic driver of that partnership.
How does your own therapeutic development fit into these partnerships? Are you doing work outside of them? Are you planning on possibly pursuing molecules partners pass on?
Yeah, so first of all, as I mentioned, we want to be a fully integrated company. So our goal is to put into the market Kymera's drugs. So actually, most of the work that we do is outside of those partnerships. And For example, we have 2 more programs that are about to enter the clinic. One is KT-413, which is actually a dual degrader. We can talk about what that means for oncology. Then we have another degrader asset in oncology, which is called KT-333. This is to go against, for the first time, again, also here, another first for Kymera in TPD was for the first time a heterobifunctional degrader against an undrugged transcription factor, which is a class of protein that are very difficult to drug. So also that is going into the clinic soon. Again, and this is going to be developed by Kymera 100% in a series of liquid and solid tumors. We have many other programs internally that we haven't disclosed. I believe we plan to disclose at least one new one before the end of the year. And we have a broad pipeline of programs that are, again, all owned by Kymera that we plan to advance. So I would say, generally, you should look at Kymera as a drug discovery, development, and eventually commercialization company with some partnerships in some programs in our pipeline, but not the other way around.
You raised $257 million in upsized offering in July. How's that money being used, and how far will it take you?
Thank you. That's a great question. So yeah, we now have, at least in July, when we closed the most recent financing, we had about $650 million in the bank, which for a company that went public just about a year ago is a very healthy balance sheet. And our goal with the money, which will take us into kind of 2025, we believe at this point, is to continue to invest in our pipeline, the one that there is or about to go into the clinic. Importantly, continue to invest in our discovery engine to be, 5 years from today, having multiple programs in the clinic at different stage, and also continue to invest in our platform. We have very unique investments in advancing the protein degradation and going into spaces that really no one has been able to do, using novel E3 ligases that are selective for particular tissues or particular cell types. So I would say the money allows us to strategically invest in areas that probably, most companies wouldn't be able to invest so early, so that we actually can build as quickly as possible a de-risked pipeline that will allow us to ask many different clinical questions and hopefully generate high-value medicines for patients.
Nello Mainolfi, co-founder, president, and CEO of Kymera. Nello, thanks so much for your time today.
Thanks, Danny. This was really fun.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.